We move on now from the preparation of the specimens to the actual data collection, and we'll have in this session 2 talks about instrumentation. The first one will be presented by J Plitzko.
Thank you very much for the introduction. Can everybody hear me, or is it okay? Fine. So when Henning asked me to give a presentation about instrumentation, I didn't know what to expect. Actually, what we are doing in Martinsried, in the Department of Molecular Biochemistry, headed by Wolfgang Baumeister, we are doing tomography. And cryo-electron tomography is a little bit different than cryo-electron crystallography. However, we are using the same instruments. And what you see here is the Titan cryos. Actually, that will be good. Thank you. That will be also one of the microscopes you will lay hands on or see demonstrations on it. And when I made that sketch, and it's approximately 4 years old when I did that sketch. So it was at that time not clear how the microscope will be surrounded by a box. But you will see in the course of my talk a couple of slides around the Titan cryos. If you have any questions during the talk, please ask. I don't know the expertise and the level of expertise I have here gathered in the audience. So if I'm just too simple or too easy, just stop me, ask me. Or if I'm too complicated, just ask again. So it should be an open talk. So this is the Max Planck Institute in Martinsried. So actually what you can see here is the whole campus. There are 1,000 people working there. And it's one of the largest institutes of the Max Planck Society. It's close to the border of Munich. So this is the border of Munich, where you can see here the facilities of the university. And our department here is nicely seated across to the tennis courts and the soccer field, which is essential because microscopists, especially electron microscopists, they are working in the dark. And working in the dark means that's the little arrow here. That's pointing to a building, which will be the topic of my teaser movie, which is called The Bunker. And the bunker is from former times when Walter Hoppe was director of that department. He built that bunker, where he put in first his X-ray machines and then the first electron microscopes to do tomography. For those of you who never have seen Wolfgang, because he is traveling a lot, and even some people in my department haven't seen him that often, I wanted to show you this picture. Just in case you meet him at the airport, that you see that this is Wolfgang who is heading that department. So that's my teaser movie. It's adapted from The Power of 10, which is very well known in Europe, but not so well known in the States. So we can also say Google Earth. So you're approaching from a large scale, from 10 to the power of 7, to a very, very small scale, 10 to the power of -7. So you miss our department here, and we go to that building, which is the entrance to the bunker. And when we did that movie, we featured the Polara microscope, but there you can see that's actually a microscope. It's not only a box. So we are entering now the goniometer stage, the compute stage, where in this case you have cartridges to hold your grid, the 3 millimeter disc. In this case, for tomography is suitable, so with long grid bars. And then you see here, you see a vitrified specimen, plunge frozen, with cells on it. Here you can see one cell, and we are approaching now almost the resolution limit what we have in tomography. See, that's the cell. And now we would like to depict all the proteins inside the cell. Like, for example, this little guy here, this is the 20S. And in the end, we would like to compare that structure, of course, or fit that structure with the X-ray structure. In this case, the 20S proteasome is very well known. And but you would like to see it at work in the cell. So no purification, no isolation procedures, just the cell, then do tomography, and then do the whole task. And that's the reason why we also use the cryo-electron microscope. Just only to give you an idea how everything is done, because I guess this is the first talk where hands-on something is shown. So this is like how we freeze in cells. So it's the guillotine-like apparatus, and at the tip, here you can't see the 3 millimeter grid, which held with the inverted tweezer. You apply 3 to 4 microliter a droplet of solution of cells or purified proteins. Then you blot away, because 3 to 4 microliters would be too thick for the transmission electron microscope, and then you let it plunge. Well, it's pretty fast, but that's how we freeze it in. That's a common standard side entry holder. This is an old movie, but I just only show you how it's done in former times, and still it's done. So you just only mount that grid at liquid nitrogen temperature from your storage box into the tip of the holder. Then you clamp it with a ring, and then you insert it in either a small microscope, like in this case a 120 kV biofilter, and then you can tilt the holder, the cryo holder, with the CompuStage in the microscope for tomography or for any other tasks. But you have to keep the sample at liquid nitrogen temperature all the time. Just to give you a small idea of the history of electron microscopy, maybe some of you know everything, so it's boring, but for some of you maybe it's new. And this is like when it started with J Thomson, with the discovery of the electron, already in the late 1900s. When Louis de Broglie actually identified the wavelengths of the electron, Ruska didn't know about it. So Ruska just only built a microscope without not knowing that there is a wave character to the electron. He just built it, and he did his PhD thesis in 29 of the 20th century, and then the first microscopes appeared in the 30s. So in the US, the Metropolitan Vickers actually built the first industrial electron microscope, and Börries and Ruska, they produced the first practical one with Siemens with a 10 nanometer resolution already at that time. And Ruska, his brother was working in biology, and so they put in samples, biological samples. And when you think about electrons traveling in air, they can't go far. So you have to be in vacuum, and you have to put your samples into the vacuum as well. So you need to have means of preparation. Dehydration, for example, was one of the means to get the sample into the microscope, but that's not good because you will collapse the structures and you will alter the structures, so we create artifacts. So you need other means of preparation techniques, which appeared in the 80s, which I haven't put here, with the cryopreparation initiated in the whole field, so that we can't just only name one person, but actually it was pioneered a lot by Jacques Dubochet working at the EMBL. And in 86, everybody got the Nobel Prize for their endeavors. So actually here in 74, already the first energy filter appeared, and I'm not talking about energy filters. If you would like to know more, just ask me later. Why do we do microscopy with electrons? So if you know about the wavelengths of electrons, then, and you know the wavelengths of visible light, you know visible light has a wavelength 400 to 800 nanometer, and you get a resolution of almost 200 nanometers. Or if you use now the new microscopy techniques in light microscopy, like STAT-PALM, like the 4 acronym light microscopy, you get higher resolutions. However, but if you just only calculate it with this wavelength, you will get 200 nanometers. So this is the Rayleigh criterion, and you can just only adapt it. You just, in terms of numerical aperture, you use the collection semi-angle, and for lambda you use the wavelength of the electron, which is dependent on the energy you're using. So if you go from 20 kV initial energy to 1000 kV, you decrease the wavelengths, and you also increase the speed of the electrons. So at 300 kV, which we at the moment routinely use, you have a speed of the electrons of 2/3 of the speed of light, so pretty fast. But that's, in principle, the most practical approach to use 300 kV. If you look at the 1000 kV microscopes, they are 3 stories high, there are lots of services required, and it's not really feasible to place them in any lab environment. But 300 kV machines are quite convenient to place them in any lab environment. So with the common transmission electron microscopes, you can resolve, and in material scientists, they just only try to resolve more and more. You can resolve down to 1 angstrom or below even. So you have a pretty good resolution with the electrons. However, I talked about that it's 2/3 of the speed of light. You can imagine if you are sitting in a microscope and you are hit by an electron traveling 2/3 of the speed of light, it's pretty harsh. And so you have to take into account that you create with the electrons some damage to your sample. just to give you a little idea how it looked in the former times, and it's pretty much the same setup now today. In former times, they already had boxes in the back of the microscope, so this is not a new idea from FEI to build a box around the microscope. They had the boxes to cover all the vacuum system and electronics in the back. But in principle, it's the same setup for every microscope nowadays, just they look a little bit simpler in former times because they were not computer controlled and you had not the whole electrical wiring to the microscope. You just had pumps and a column. But in the column, you had the same lenses as nowadays, just only a little bit more sophisticated nowadays. So this is just the Polara I showed you in my teaser movie. And this is a special microscope which was developed in the late 90s and in the first part of the 21st century. So you see here, this is the normal column. And in the next slide, you will see depicted where the lenses are. You see a big dewar on the right side. And this dewar is filled with liquid nitrogen to cool down the sample at all times. So you only have to refill the dewar once a day. And the sample stays at liquid nitrogen temperature for 24 hours before you have to refill the dewar. The advantage with this microscope is you can also fill in liquid helium, so you can cool down even further that microscope to approximately 10 Kelvin. So that's a quite low temperature. Why would you use a lower temperature? Because it's suspected that if you're going lower in temperature, you decrease a little bit the time. You increase a little bit the time you can investigate your sample. So you just only have a more resistant sample at lower temperature. But that's not really true because the ice changes at that temperature quite dramatically. So you lose contrast a lot, and that's not very, very feasible for tomography. For our cases, it is feasible for crystallography, where we have very, very thin layers of 2D crystals. There, you can really do a lot of good work, and that's done in Fujiyoshi's lab with GL microscopes, so not with this type of microscope. However, this microscope first featured a stage where you had 6 samples to put into the microscope, and the transfer was done not via air, but via vacuum. So you can put in 6 samples at a time, and you transfer them in vacuum, which makes the approach to the microscope easier, because you have to breach at one point the vacuum of the microscope, and that's always the critical part where you get contamination. With a side entry, you had to do it manually, and it takes a lot of training to get people adapted to doing the insertion of the side entry with the microscope. And it was completely computer controlled at that time as well. So in principle, when you look at the Titan Cryos, it's not that different from this microscope. It's just a box around it where you can't see inside, and it's very, very difficult to take photos of inside because it's crowded. So this is how the microscope is set up. So you have, in principle, the same setup as in the light microscope. There you have the gun where you accelerate the electrons to 300 kV in this case. You have a condenser lens system. So normally you have 2 condenser lens systems. In the Titan you have 3 to get constant parallel illumination over all magnifications and all illumination conditions. You have an objective area, where in this case you have the objective aperture here, and you have the projection chamber and the projection lens system. So you have 3 systems, and you have also 3 systems of lenses and 3 systems of coils. And these coils, these beam deflecting coils, are very, very essential. And Hans Herbert, when he talks about alignment, he will talk a little bit about these coils, I guess, because they are very, very essential in all the alignment and tuning steps of the microscope. So this is just only depicting where the specimen sits, where the coils are. One are above the specimen, one below, which are very essential for low-dose exposure schemes. And you have the condenser setup where you just only increase the illumination when you would like to look at your sample. When you interact with your sample, I told you that you create maybe some damage, but how is this damage generated? So first of all, you have the electron beam hitting the sample. Then you create a lot of interactions which you can use for scanning electron microscopy, like, for example, the backscattered electrons with the backscattered electron detector or secondary electrons. You can also use the X-rays for elemental analysis for the heavy atoms. You can use the Auger electrons, or you can use the cathodoluminescence. So you can analyze the sample on the surface with the SEM. In former times, the transmission electron microscopes, they just only had also the SEM capabilities. Nowadays, that was skipped. Or you go through the sample in the transmission mode, and you get two types of scattered electrons. The elastically scattered electrons, which are very, very essential for the forming of the image contrast, and the inelastically scattered electrons, which are unwanted. Unwanted for the imaging case. If you want to do analytical electron microscopy, you use them, because they can be used for electron energy loss spectroscopy to analyze what are the chemical constitutions of your sample, or they can be used for electron spectroscopic imaging, just to need to do nice elemental maps. And you have a fraction of a bulk fraction, if the sample is thin enough, of unscattered electrons. You have to remember that these elastically scattered electrons, they are in a range of 10 degrees, so a large area, angular area, which are covered, so you can block them partly with an objective aperture. But the inelastic scattered electrons, they are forward scattered, so they are in a very narrow range, angular range. So they are always, they can't block them with an aperture. You have to use some special devices, and that's called an energy filter. So you just only filter them out by using a magnetic prism. So that's just only how the elastically scattered electrons, they are just like billiard balls against the wall. They change the direction, but they do not transfer per se energy. In the inelastically case, you have a crash test, you hit an electron of a sample atom, you knock it out, and of course by knocking it out you lose energy and you get an inelastic scattered electron, which in this case causes beam damage because it deposits energy into your sample. That's how it looks like when you illuminate, for example, one cell on a lazy carbon grid in ice with a dose of 50 electrons per square angstrom. And that dose, 50 electrons per square angstrom, is equivalent in principle, when you calculate it in meanings of gray in radiochemistry, to a dose of 1.6 to 10 to 8 gray, which means that's normally a dose which can be observed in the vicinity of a nuclear reaction. So it's a pretty high dose with 50 electrons per square angstrom at 300 keV. When you increase the dose by 10 times, then you create the melting of the ice, of course, the bubbling, because you are forming radicals in the sample, and they are agglomerating to gas bubbles, and then at one point they get so big that they destroy your sample. So in this case, everything is gone, so you can't use it anymore. And with 50 electrons, you stay actually below the visible radiation damage. It's not like that you do not create radiation damage when you just only use, for example, only 2 electrons per square angstrom. You create some radiation damage, but you can't see it because you maybe break one bond, but it will not change a lot of things. So you have to stay, in principle, what's now termed as the maximum tolerable dose is like 50 electrons per square angstroms for tomography when you do a whole tilt series, so hundreds of images. Or when you do single particle, you just only stay below 20 electrons per square angstrom because the resolution you would like to achieve in single particle analysis is much higher. And if you now do crystallography, and I would ask Henning, because I don't know the doses which you normally use, you might stay at least 10 or below 10. So, I guess it's depending on the resolution, how much dose you can spend on your sample. Because the resolution you would like to get in the end on your reconstructed molecule or on your reconstructed cell. In tomography, you have a resolution normally routinely of 4 nanometer, so it's not in the angstrom regime. And for single particle, you can get below 10 when you collect enough particles. When you have higher symmetries, then you can also go, like Hongzhu showed, like with virus particles, you can go to 3.5, 3.8 angstrom. But that's exactly the same resolution you would like to have also in membrane proteins when you crystallize them in 2D arrays. Talking about low dose, you need schemes to operate a microscope so that every tuning, focusing step, alignment step is done in an automated way, and that's done with the image beam shift system. And I guess also Hans will a little bit elaborate on the image beam shift system. So what you do is you would like to shift your sample away from the beam or the beam away from the sample. But you would like to keep the beam always in the optical axis. And so that's done with the two, where are they? They are gone. So they are gone from the slide, so I don't know where they are. So actually, at this slide, there were these beam deflecting, oh, there are beam deflecting coils here, and the image deflecting coils. They have to work in a concerted fashion, so to keep the beam centered at all times. When you move the sample, like with the image beam coils, the beam stays put, and when you shift the beam, the image stays put. And when you, for example, doing low dose, this is just a low dose acquisition scheme of tomography, but everything is in principle the same when you do cryo. In principle, the tracking step is you exchange by a search step when you do cryo. When you do the searching, you do it in low magnifications, and then you go up higher in magnification. Then you would like to focus, but the focusing you would like not to do on your sample position, because you would extend the time at that sample position too much, and you would require lots of doses. And that means you have to do it adjacent to your sample position. So you have to shift, and you have to stay always in the optical axis. And then you can do the exposure at the final position. So this low-dose acquisition procedure is very, very common, and it's so-called the first-generation software, which you have to operate the microscope with. Of course, the whole procedure was feasible first with the invention of CCD cameras, digital detectors, because then you can assess the images directly. When you have negative plates, it's a little bit more tricky, or by eye, it's also a little bit more tricky. So I talked a little bit about this stuff here, and I will not talk to CCDs and camera detecting systems. Andreas Engel will elaborate on that in the next talk. And the energy filters are only used for thick samples, for cellular tomography, for example. They are not used in crystallography or in single particle, because the thinner the samples, the less inelastic scattered electrons you create. And that depends on the mean free path, which I'm not going into it. When you look at the microscope, and this is just like a microscope with an oil diffusion pump, so you have the vacuum system of the microscope here, depicted like in a normal Polara case. And you have several pumps. You have the pre-vac pump, you have the ODP, the oil diffusion pump, the turbomolecular pump, and the IGPs. But oil diffusion, when you hear about oil diffusion, you just only think about contaminating your column with oil, which is true, because you diffuse oil to suck out all the molecules in the column. So oil diffusion is not really wanted, because it will contaminate at one point your sample. The turbopumps are generally much better, because they're just only at a high spin. They remove the molecules from the sample, and the pre-vacuum pumps are necessary to establish just a little bit of a vacuum in the column. Ion gather pumps are normally used for the gun area and the direct column area to pump by ionizing molecules from and remove them from the column. And you have to remember, ion gather pumps, while the turbo pump is pumping the molecules out of the microscope, the pre-vac pump is pumping the molecules out of the microscope. The ion gather pumps, they trap the molecules in the microscope. So when you heat up, for example, the ion gather pumps at one point, you will create lots of molecules in your column, because you have trapped them first, and then they are given back to the column. And you might wonder why I tell you this, because every one of you will use the term cryocycle at the microscope. Press a button, and what does the cryocycle do? The cryocycle does not, it's very, very simple. It shuts down the ion gather pumps to heat them up, and it activates the turbopumps, because the turbopumps, they can remove the molecules out from the microscope. Ion gather pumps, they just only let the molecules stick to the magnets. So to remove all the contaminants out of the microscope, you have to use the cryocycle, and during a session at the microscope, you create lots of vapor from at cryo experiments all the time, and hydrocarbons, and these have to be removed at one point. So after 7 days, for example, with the Polaras, you heat up the system to get every potential contaminant out of the microscope. What you have seen here, this is the typical log scale of FEI. So you can remember that log scale, or you can just only adapt it to the millibars or Pascal level, where you can see that you operate a microscope in a level, in the column level, it should be in 10 to the minus 6 millibars, where in the camera level, it's not that good, normally never. And in the gun level, it should be very, very clean, because if you contaminate your gun, you will decrease the lifetime of your fact tip, for example. You have for high resolution, that's the title of the talk, high resolution data collection. High resolution means you have the microscope, and data collection you have a camera. And but you have three transfer functions in the microscope, just only to repeat myself a little bit. You have the gun, where you have an electron spread. If you use a thermionic gun, it's different than from a field emission gun. Then you have the objective system, which is characterized by spherical and chromatic aberration and the focus you are using. And this everything, like the gun and the objective system, goes into the contrast transfer function. While the CCD is characterized by the point spread and the noise of the CCD, and that goes in the MTF and the DQE. And these three transfer functions, or these two in the end, where these two are combined, go into the resolution of your microscope. And you can imagine then when you have a CCD with lots of noise, you can't see anything. When you have a CCD with a point spread, which is like blurring your image, then you can't see anything. So it decreases the resolution a lot. So you have to take, you can't just only buy a microscope, you have to buy the CCD as well. Or you have to use negative plates. This is how it looks like with thermionic guns and field emission guns. So you have, in the thermionic case, you use a crystal, you heat up the crystal, and the crystal emits electrons. In the field emission case, you have a very, very sharp tip. It's normally a tungsten tip coated with zirconium oxide. And you can imagine when you just only get contaminants on a hot tip, then you will destroy that zirconium oxide coating of the tungsten, and then you will create a damaged fact tip, or you will just only lose the fact tip completely. So that's the reason why the vacuum in the gun area has to be very, very good, and you have to remove all the contaminants from your sample. That's how a lens looks like. So you have a coils, circular coils running, and the electrons are going through these coils in circles as well. And when you, for example, align the rotation center or the stigmatism, then you will see that you have like these circles where the electrons will travel through the column. So you create a magnetic field and they travel through, and this is just only depicting of the optical axis. This is how coils look like in real nature. So when you operate a facility with electron microscopes, you have, at one point, you have to disassemble them and service them. For example, when you drop a cartridge in the Polara. So this is how it looks like when the Polara is split. This is the objective, and also it's called the octagon because it had 8 ports. So you can attach an objective aperture, attach a holder, attach the vacuum system, attach an EDX detector or whatever. So you have 8 ports where you can put something in. And in this case, it was just like, this is the upper part of the objective, and this is the lower part of the objective. And you see the blue box here, and that's the casing where the lens is cooled, because you can imagine if you heat up a lens with milliamps, then you will create heat, and the heat has to be removed, and so you have to have a cooling system of the microscope. This is just only, you see a little bit, and you see the cup here, and that's the actual lens. It's not, the cooling system is here, and that's the cap, and inside the cooling system you have the coils, the magnetic coils. This is how a coil looks like when it's broken. So you can see a crack here, and you see the plugs for the copper coils. So you put in just only normal voltages here, and you get milliamps for creating the magnetic field inside here. And so when this casing, where the liquid, the cooling liquid, is floating, and this casing cracks, you will have water directly in contact with electricity, which is not very good. But that's normal. That happens sometimes. And so you see here a plastic casing, but thanks God, plastic casings are not any longer in fashion. So you're using metal casings, so which can't normally crack like this. So that's like some of the optical aberrations of the lenses. I'm not going into detail into it. I just only want to show you spherical means that you just only displace in a disk. So lateral displacements. Chromical means also lateral displacements, but depending on the energy. So normally you have a CS, a spherical aberration constant of 2 millimeters or a little bit more, and CC in the same range. That's how it looks like when you have the objective lens active. So when you take an image of particles at 0 defocus, for example, then you increase the focus and you see the particles much better because you increase the low frequency information. Here you can see the corresponding FFT, and you can calculate how much transfer you will get with the contrast transfer function. And that's in principle just only here. That's the defocus term. And that's the term which is pretty much fixed by the wavelength and the spherical aberration of your objective lens system. So with that one, you calculate the phase contrast transfer function, and you get these nice oscillating patterns when you, in case you use a field emission gun. You have these wiggles, and you have a dampening over spatial frequencies. Ideally, it would be a box, so you have contrast transfer up to 1, so black and white, and that would be perfect, but you don't have it. You have a decrease, a damping, and that spatial damping, that determines your information transfer. So now we go to the new stuff. So what's the time now? How much time do I have left? 10 minutes, good. So speed up. So I gave a talk a couple of months ago, and I termed that talk the goodies from Pandora's box, because actually the Titan is a box. It's a black box for some people. And you know a little bit about Pandora, because Pandora actually was related to the Titans and the other cryo gods. And when you go into the Wikipedia, you just only look at Pandora's box, and that means it's a negative term, which is about the unanticipated consequences of technical and scientific development. And when you face the Titan cryos, you sometimes think, well, why did we have to open that Pandora's box? And when you look into the history of Pandora, actually Pandora was told not to open the box, but she was too curious to open it, and she opened it. And every evil came out, but she shut the box in time, and the last thing what remained in that box was hope. Actually, but the time being when she reopened the box was a lot of worries in the world, because the world remained extremely bleak for an unspecified interval during the time when she first opened the box. But when she second opened the box, hope flooded out, and then everything was good. So actually, when we opened the Pandora's box 4 years ago, it was really, the unspecified interval was 3 years. It was extremely bleak for a couple of years, and now we have hope that everything is going back and everything is going to normal. Well, Cryos is also one of the 4 Titan. So, well, actually, there were six brothers, and one was termed Cryos, the pillar of the south from the cosmic pillars. I don't know about the Greek mythology, so I'm sorry. But it's also the name for the next generation of TEMs from FEI. So you have low-base Titans like you see here, or a Titan Cryos like you see here. And how it's installed in our lab, which was the first prototype, and how it's installed here in Henning's lab. And you can see it's a high base system, so you have a 4-point suspension system where you have sitting the column inside. You have a special loading device, which is called the autoloader. You have a gun, and the rest is the same thing as a normal microscope. You have specific coils. You can do completed automated and robotic sample transfer. It's automated in cooling, so you can leave the microscope for 7 days, 10 days in a row if you want, if you have a large enough viewer to supply the microscope. You can operate, and that's the neat thing, the microscope at different voltages very easily. So the optics are very stable. So you can just only tune down to 80 kV, or you go up to 300 kV. And in this case, you have also dual axis capabilities with the stage, which are handy not only if you do tomography, but also if you do, for example, crystallography, because you would like to tilt your samples in the right zone axis, for example. When you are doing material science, it's very, 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 very suitable. This is how it looks like in the case of the MPIV. So we have digged the hole to accommodate a 4 meters high Titan, and we have made a computer control where you can process the data online at the microscope. And you have the control computer of the microscope sitting here, which looks here, and it's running our own software to control it. That's how it looks like when you do a large area detection with an 8K detector. So that would be equivalent to a 4K image. And that's how it looks like in a high dose, high defocus. And that's the normal image you get when you are looking at single particles. You don't see the particles, but you need a high dose to get the particles out of it and to align them. The autoloader has several components. It was developed in a project called Interaction Proteome within 6 framework. It has, well, this is the column of the microscope. Then it's attached, well, this is the CompuStage area where you attach normally the sample to tilt it in X, Y, and Z, to move it in X, Y, Z, and to tilt it in alpha. or in beta. Then you have the cooling system, which is very important because if you have temperature leaks, you get a very, very cold surface at one point, a very, very warm surface at the other point, and then molecules can go from there to there where it's colder or warmer. And you have to be very, very careful when you design such a cryo box around the sample. Then you have the autoloader, which is a two-arm system. You will see it in a minute. And of course, you have a dewar to cool down with liquid nitrogen. Then you have a capsule which is filled with liquid nitrogen, where you put in a cassette with 12 grids in liquid. So you don't transfer in air, you transfer in liquid. And then you have a manual loading station, which is the bottleneck. So you have to fill in 12 grids into that cassette, and you have to put the cassette into the liquid of the capsule, which is not very well done at the moment. That's how it looks like in the cut drawing. When you cut it open, you see the two arms. You have a gripper, and you have for the outer grids, which you will see in a minute, so special grids, reinforced rings with a normal copper grid. Then you have a cassette gripper to move the cassette up and down, and you have the capsule here attached to the microscope. That's how it, to the autoloader. That's how it looks like in real life, so it's a lot of wiring. And that's how the outer grid looks like. So it's a reinforced ring, which is 0.4 millimeters high and a little bit larger than the normal 3 millimeter grid to accommodate a 3 millimeter grid. And then you have a clip ring, which you can insert into it to clamp the grid tight into the outer grid. It's the smallest possible holder solution. It's just a cartridge, but it's very, very small cartridge. When you look in the microscope, so the outer grid is transferred to the holder, and the holder has two axes. And these two axes, they can move and doing the 90 degrees rotation. So if you would like to adjust to a specific angle in beta, then you can do it with these two arms. And then you can do, in this case, dual axis tomography. So you just only tilt 90 degrees and then do the second take of for a single axis acquisition twice, but 90 degrees rotated. And then you do the same thing again. This is pretty neat. And when you use your own software, you can accommodate beta tilts of different degrees. When you just only use FEI software, you can just only say 90 degrees plus a little bit, but 0 or 90 degrees, so you have a discrete tilting availability. That's the transfer station and the cassette, how it looks like. So you see it's a lot of ice contamination here, and it's not a very, very nice transfer. And you can do actually good things with the microscope. So that's what we have tried in the beginning of this year to benchmark the microscope, because the idea is to have a high throughput base. So you would like not to sit in front of the microscope and collect thousands of images to get the particles for single particle investigations or batch tomography. You would like to let the microscope run. You have 12 grids in there, and then let it run, come back in 12 days or in a month, and then you get the data and you analyze the data. Or you do the analysis of the data on the fly. So the structure of the 20S, so that's the 20S. So this is the 20S on the scale. It's 700 kilodalton in weight and 50 nanometer tall and 11 nanometer in width. That's the proteasome 20S. That's how it looks like in the PDB database. That's how it looks like in negative stains average particles. That's how it looks like when you take the PDB structure and render it to 10 nanometer resolution or even to less nanometer resolution. So it's a ton. It's a trash can, so to speak. And it's, of course, it's a trash can of the cell. That's how it looks like when you take top views or side views of the 20S. And that's just the negative stain representation, just to give you an idea how this molecule looks like. That's how it looks like on an 8K image in a nice homogeneous ice layer. If you zoom in, you can see all the top views and the side views. And then you can collect data. Oh, that's nice. In this case, that's the EM map derived out of 50,000 particles collected in 24 hours. So 50,000 particles, that means 1,200 image pairs. So 2,400 images automatically collected. And out of these 2,400 images, you have, of course, focal pairs. So you have two types of foci. and one high focus to align the particles and to pick the particles, and one low to get the resolution. And 50,000 particles out of 1,200 images in 24 hours, and that can compare nicely with the crystal structure rendered at 7 angstroms. So you just only don't need a comparison here, or just only a Fourier shell correlation. You can see that you have the resolution when you collect the data, and you have enough symmetry. In this case, you have a 2-fold plus a 7-fold symmetry, so 14-fold symmetrized. So in one day, you can, with 50,000 particles, get to a structure which is below 10 angstrom, which is nice. But if you have no symmetry, then 24 hours is way too less. So you need more time at the microscope, and that's the reason why you need a high throughput pipeline. And that's just only the comparison when you fit in the PDB structure. Of course, for this endeavor, you need software, and we have just only programmed to the microscope a software which is called Tom, and Tom is suitable for running the microscope in a complete autonomous fashion. And I'm not talking about the software. I talked about the instrumentation. I talked about a little bit about what's the critical parts of it. And I think I'm now almost finished with the time, and I stop here before I go into details with the software. Thanks a lot.
Thank you. We have time for 1 or 2 questions.
So very nice. The comparison of the 7 angstrom AMAP with the X-ray structure rendered to same resolution, they look quite similar, but there were significant differences.
There are differences, yes. There are differences.
Why is that? Is that are there different states, or there's a bit of helix sticking out at the side?
Actually there are some areas are sticking out here, which is known from EEM structures. When you compare it, for example, from Ifan's structure, Ifan Cheng's structure, then you can see he has the same areas. But well, we can't just only say for sure, but they are similar. But in some cases, they are a little bit different. Definitely.
Do you know why they're different?
No.
Other questions?
You had a Titan scheme that showed, I think, the helium stage, wasn't it?
Yes.
Okay.
But helium is off the books now. So.
Yeah. Why?
Actually, I showed you the instrumentation, but I didn't talk too much about the problems. I just only said it was Pandora's box for 3 years, and I just didn't elaborate on what kind of problems you have with the Titan, or what you had with the Titan. I have to rephrase. So you have the autoloader, this robotic arm. That's one of the major issues. It's not quite stable and robust, so there are some reliability questions which have been fixed over the last 3 years. And the second thing is the contamination during investigations. So for example, I would be happy to show you, instead of a 20S, a 26S, where you have only a 2-fold symmetry and you would not like to symmetrize. But you need also like 750,000 or 1 million particles, but you can't collect them in 24 hours. You have to collect them in weeks or months. But one sample in the microscope in former times only stayed contamination-free for 24 hours. After 24 hours, you had already collected enough contaminants on the top that you were blurring out the particles in the ice. And so you couldn't collect data for an extended period of time on one grid. And that's a major issue with the cryo box I mentioned at one point. It's the temperature around the sample. So one of the arms were not right in temperature, and so you had to investigate which of these arms were too warm to not drag away the contaminants, but just only to give the contaminants back to the sample. Also, there were some problems with the seals of the microscope. There were plastic seals instead of metal seals. They have exchanged them. So now everything is going a little bit better. And the goal is to have a contamination rate, and you can just only say approximately. I can't give you real numbers. It's 0.2 nanometers per hour. That's acceptable. then you can operate a microscope at least on one sample for 3 days. And it's different if you operate a microscope in the high throughput fashion, where you collect 2,000 images and do all the alignment steps with the focusing and tracking and so on. Or if you just only collect in the morning one image, in the afternoon one image, and in the evening an image. If you don't do anything with the microscope, you do not create any vapor arising from your sample. Then you don't create any contaminants. You create contaminants from the outside, which build up, but not that much as when you operate a microscope in a high throughput fashion. And that was investigated over the last months quite seriously, and now we have solutions to cope with that problems, and the new version of the Titan is equipped with that stuff. Okay, the question why it's not helium temperature, because it's another problematic issue added to the whole thing, because helium requires much more into this temperature stability than liquid nitrogen. And in the Polara case, it was designed by Gatan, Oxford Instruments and Gatan. And in the Titan case, everything was done exclusively at FEI, and they don't have really the helium experience, so to speak. This was planned, and it's just, I guess, it's only postponed, that they just only maybe they will come up with a new strategy and R&D in helium. But I guess at the moment they would like to have a reliable Titan first before they go into helium.
Yeah. So. Has anybody tested the cryo system with 2D crystals diffracting at high resolution? How well does the instrument compare in terms of stability, in terms of data collection to let's say, to a GOL 3200?
That's a good question. I can't comment. I'm not a GOL user. I guess Hans is more familiar with GOLs, and also Henning from former times. I'm not really able to give you a statement which is better. I can just only tell you that, and of course Werner is also a GOL user. The thing is that you have, the optics are very, very stable. That's quite clear. So you don't, if you switch magnifications, if you do some changes in the illumination system, the hysteresis is very, very, very low. So you can switch very fast, and that's good. It's also the same thing with the stage. So movements are much easier compensated. So that's a bonus. And you get higher resolution when you do just only normal 2D imaging. And also with our system, when you just only do a test, then you can get 1.2, 1.3 angstrom when you do the resolution test of the microscope. Very, very easily. It's not that you have to fiddle around a lot with the microscope. And that's a big bonus. But a comparison between two microscopes, if you do a comparison like this, it's difficult because it's a different sample, because cryo samples are different. It's just like a real comparison. I don't think that it's really feasible or really wanted. But it should be done by the companies, not by scientists. But I guess GOL is not very interested in comparing the microscope, and FEI is not very interested in comparing the microscope.